Ellie Hummel, and this year my project is exploring the effect of EML-1 on axon growth. The key to finding treatments to resolving spinal cord injuries lies in understanding plasticity in the neuron. I believe one solution might be EML-1. EML-1 is a gene that is commonly activated in early brain development. It is associated with the growth of neuronal progenitor cells in the early brain and is crucial to have a healthy development as a child. So the first purpose of my project was to see if EML-1 was affecting axon growth, and I did this last year by using PC12 cells, which are polychroma 12 cells, and they actually just come from the adrenal gland of a rat, and they can be differentiated with NGF which is just nerve growth factor, to become neuron-like. So after you differentiate them with NGF, you can run experiments on them like they're neurons. So last year I treated them with EML-1 siRNA, and I saw the effect it had on their growth. I found that EML-1 was a positive upregulator of collateral sprouting. This was really cool, and so I was super excited. But last year I didn't have time to make sure that the siRNA type I was using was validated and actually the correct type, because I did my project rather quickly. I only did one PCR, and I don't believe you can see it from the video screen, but if you look here, there's 2 bands in the EML-1 PCR. So this signifies that there's a non-specific band, and that means that if in my experiment, I'm not sure if the non-specific band is being activated or if EML-1 is being activated, which was actually supposed to be attached by my primer that I developed. So I was concerned, so this year I decided I wanted to validate the siRNA type I was using and ensure that EML-1 was actually being expressed in the cells that were collaterally sprouting. I also wanted to see if I potentially needed to redo my project from last year to make sure that my findings were actually accurate and they weren't just a fluke due to quick experimentation without repetition. So I hypothesized that EML-1 will continue to act as a positive upregulator of collateral sprouting because of my previous research last year, and because of its early role in brain development and its presence in neurons as protein. So my second hypothesis was that EML-1 siRNA will continue to be the most potent siRNA. This is because I saw the effect I was expecting to see last year, and also it appeared to be the most effective siRNA type in the PCR I ran previously. However, I was not sure of this, which was again why I wanted to do this experiment this year. So my third hypothesis was that EML-1 would be present in the differentiated PC12 cells after hybridization, which basically just shows what genes are present in the specific cell. So I continued into my methodology and actually implemented my project. And again, this year I started out with a lot of PCRs, which is just polymerase chain reaction. And to do my PCRs, I developed primers which just attached to I did reverse transcription PCR, so it's basically just PCRs with RNA, but you add an extra enzyme called reverse transcriptionase, which allows the PCR to be done with siRNA. With RNA. So I did this, and I saw in my PCRs that I still had a double band. This was the first one I ran this year, and instead of doing a 1% agarose gel like I did last time, I did a 2% agarose gel on EML-1 and GAP-DH, which was my control. And this gel was also inconclusive as seen by the double band, so this helped me realize that different concentrations of agarose, although it did improve the image quality, did not necessarily affect the double band. So this kind of rules out the fact that the PCR was run incorrectly, though I did do several more tests. And in this one, I again used the 10% agarose gel because of the improved image quality on the last one. And I also used new cDNA to eliminate the chance of contamination of my last cDNA that I was using for the reverse transcription PCR. Again, it was inconclusive. There was a double band, so I couldn't conclude whether or not the siRNA type was the most effective. So then I did another PCR, And this time I did a 2.5 concentration of agarose to see again if the concentration had anything to do with it, and potentially 2% was just not high enough. And I used cold TAE, which is what you put the gel in to have it solidify, and a larger concentration of agarose again. Again, the PCR was inconclusive because of the double band, although the image quality did improve. This was a very nice gel. It was probably the prettiest I had. Not that that's important. And then in my last PCR I did a 2% agarose gel again, and I actually got new stock. So I took the cDNA directly from the rat, and to make sure the stock I was using for my cDNA wasn't contaminated. And again, the gel was inconclusive. So I couldn't define from these PCRs, which I ran all the conditions more than once. I just don't have all the pictures up there because there's a lot of them. So I couldn't tell from any of my PCRs whether siRNA-1 was actually the most effective type. So I decided I was going to do something else, and I used PC12 cells again to simulate neurons, and I treated them with all the different siRNA types to see which one had the largest concentration of differentiated cells. To quantify the cell's growth, I used a program called ImageJ and staining techniques, using the stains Texas Red and DAPI, which is a nuclear stain, and Texas Red is just like a process stain. What I found was that siRNA-1 had the proportion of differentiated cells was 65%, while in the control it was only 31%. So the siRNA number 1 had 34% more differentiated cells than the control. siRNA-2 also had less differentiated cells than siRNA-1 at 54% differentiated, and siRNA-3 had less than siRNA-1 and 2 at 45% differentiated. The two-tailed p-value for the difference between the control and siRNA-1 was significant at 0.0001, which is incredibly statistically significant. So I concluded that siRNA-1 was actually the most potent siRNA type. At this point in my research, I have not completed the hybridization, which would show that EML-1 is actually present in the neuron, though hopefully I will have this done by regional science fair. So far, what I have concluded is that my hypothesis was supported, because EML-1 was in fact, siRNA-1 was in fact the best and most potent siRNA type, and this supports my findings from last year and hopefully shows me that I do not need to repeat that experiment. However, hybridization will show what I truly need to do.